And I'm waiting for the screen to come up. Or should I start the intro?
You can start the intro. I think you got to click on the screen to pull up into the full view.
Oh, okay. Very good. Because I'm just looking at you now, Jeremy. Hang on. All right. So there's Stephen. All right. Welcome, everyone, to this month's Orion webinar entitled Collaborative Bottom-Up Modeling in Computational Biomedicine. Presenter today is Stephen Larson. He's an expert on the intersection between computer technology and biological systems, and is interested in how computer systems can help us to understand fundamental principles of life. Stephen is the Chief Executive Officer of Medicell, a systems biology, informatics, and consulting company. And he previously served as the Chief Information Officer of One Mind for Research. As one of the founding partners of OpenWorm, which is not quite as squishy as it sounds, Stephen recently successfully crowdfunded the organization to the tune of $120,000, a little bit more. Congratulations again on that, Stephen. And his new green Tesla has been seen zipping around San Diego with the vanity plate iWorm lately. But you can ask about the worm and his new car in the Q&A immediately after. You can click on the button in the lower right-hand side of your screen to chime in on the Q&A. Please do so. Stephen has also worked as a professional software engineer for a major New York City investment bank, co-developed a patent, presented at more than 24 forums, published in academic journals such as Frontiers in Neuroscience and Nature, and has had his work featured on the New York Times, The Atlantic, Wired, and MSNBC.com. He holds a Bachelor's of Science and Master's of Engineering from MIT in Computer Science, as well as a PhD in Neuroscience from the University of California, San Diego. Outside of science, Stephen is one of the leading male synchronized swimmers in the world and is leading the charge to make it an Olympic sport. His best-selling cookbook, The Elegance Table, is considered the Bible of nematode cuisine. Stephen will confirm those, and if you have any questions about those, please ask in the Q&A. But we'll focus today on collaborative bottom-up modeling in computational medicine, presented by Stephen Larson. Welcome, Stephen, and enjoy the presentation, everyone.
Thank you, Lenny. Thank you very much. I think our fact checker on staff here at Medicell may have to check in on some issues with my bio, but we really appreciate the faithful reproduction there of what you were granted, so thanks for that. All right, so I'm going to go ahead and switch over here to my presentation, and thanks very much, by the way, to Orion for inviting me to get the chance to speak today. It's always a pleasure both to work with Orion and to get to speak in this forum with so many talented people working in this space. I think that there are great challenges ahead for us in biomedicine, and I think that computational technologies can only help in that challenge, and I really appreciate Orion's role in being a leading light in making that possible. So I'm going to tell you today about some of the work that we do at Medicell to try and attack this problem from a particular angle, which is the bottom-up angle, and try to do that in a way that enables multiple people to come together to help us better understand biology. So at the core, the challenge that we feel like we're facing in this field right now starts with just the simple premise that biology is complex. This picture on the left is just a small fraction of the complexity that we find in living cells, which of course are the building blocks of all of us, of practically all life on the planet. And not to mention all the challenges it scales above this and all the challenges it scales below this. And we have the tantalizing opportunity with computer systems to be able to represent things and solve problems using math that we have never been able to address in the past. And so it's very attractive for us to use this really powerful tool of computers that seem to be reaching into every corner of our lives and try and help understand not just how to make a better drug, although that's an important piece, but also to fundamentally understand disease, to understand how a biological system fails. And in order to understand how it fails, we feel that it's important to understand how it succeeds and how it manages to accomplish the complex biological functions that it carries out. So we see the challenge that is being undergone in the field of computational biology field that we sort of look to for inspiration, as first starting from this premise that biology has a certain physics associated with it. There's a physical reality of how those biological mechanisms that were shown in that picture before work. So we might have a picture like this, where the physical reality, in this case, is a picture, a cutaway picture of an axon, and we're looking through an axon and a neuron at the pieces and parts of that cell inside, so some of the organelles that live inside of this cell, like a mitochondrion, and it's wrapped up by another type of a cell which provides a sheath around it. And all of this organization, underlying it are processes that can be described by equations of physics, equations of motion very simply, or energy transfer equations. These dynamics are explicable. There are ways to understand what's going on. They are just very challenging to do so. At another scale, interactions between cells, like astrocytes interacting with glial cells that are wrapping axons in the following, also have dynamics that govern them. And additional research that looks into this does form equations that explain how the astrocyte works and how, what speed at which the myelin sheath is wrapped and what that does for the conduction velocity. And I'll show you some examples of that a little bit later. So one of the fundamental challenges in dealing with this physical reality of biology is the multiscale problem, whereby what I just showed you in the past slide is two different scales at which we can describe a biological system. In fact, the reality is that the biological system is sort of has infinite scales sort of within it that you could pick to start to model. So when you're doing this work of converting the squishy stuff into the ones and zeros, like I showed in the first slide, we sort of confront this by thinking about it as separating different scales into different boxes that we can draw boundaries around and try to work with and connect together. So like what you have in the upper corner here is that some component like this is actually made up of a bunch of subcomponents, which are themselves made up by subcomponents, and so on and so forth as you go down. And at every level here, it turns out that scientists have discovered diverse models and diverse algorithms, diverse physics, if you will, that govern the rules and principles that happen at each different scale. And all of those, although complex, are also knowable and measurable as far as we found in biology. And in addition to the spatial scales, of course, these things evolve in time. And again, the physics of biology are described in different time scales. So we have all those parts that we just mentioned that are operating independently of each other, sometimes much faster at the smaller scales and much slower at the larger scales. So all of these are challenges for us as we try to take our understanding of the physics of biology and to do something with it. So, as I mentioned, often what researchers do in the field of academia is that they take those physics principles and they describe them as equations. And for a good chunk of scientific discovery, equations have been a very powerful tool for us to describe dynamics in the world. Obviously, we got calculus, and we've done a lot of astrophysics with it, and we've built a lot of tools using formulas and systems of equations to do that. And so this is a very powerful tool as well for us to kind of get a more quantitative description of what's happening. And so this is an example of equations that are describing transitions of ion channels, for example. Okay, so to take this, though, into the 21st century, more and more often we find that in academia those papers, although a smaller fraction of them, but that those papers end up being implemented in code. Okay, so at some point those equations become source code that then allow us to play around with some of the values. That's a much more flexible thing, obviously. This is sort of where the computer enters into the equation. And so you might get a model that looks like this. This is a schematic model. I'll walk through the details of it a little bit later. But the point to take away from this at this point is that you can take those equations and turn them into traces like this that you see on the right-hand side, which are calculated by a computer crunching through those equations and plotting point after point after point that ends up in a curve. And now this is something that you can actually compare back to the original biology and see how, in this case, this is the path of an action potential as it goes through a different part of an axon that either has myelin sheaths or doesn't. So, and that's very useful for us to be able to use the computer to get answers out that we can actually compare to check and make sure that in fact the physics that we thought was happening appears to be happening when we crunch the numbers. Okay, but herein sort of is where often papers stop, and this is the challenge for us in the 21st century, is to understand how we go beyond just individual pieces of code and individual systems of equations that are described in one paper or two papers, but to actually turn this into something much larger. And so what we think about in Medicell is exactly this challenge of how do we take the best of what's being described in computational biology and how do we translate that into software that is usable by people who were not the original authors and people who want to play around with those tools, who really want to grasp the insights that are coming out but don't want to spend the time to re-implement every single paper that has ever come out there or dig around the internet for source code that might appear in multiple different languages. So what we really aim for here, and what we're involved in several collaborations to make possible, is to create maintainable software for simulating the physics of biology. So what we really want to have are software modules that interoperate and scale on a platform like this. And so what I'm going to tell you about a little bit later is one of the open source projects that we've been contributing to and helping drive the design for at Medicell, called Geppetto, and we'll tell you kind of how that helps the challenge. So ultimately, what we want is that the paper that computational biologist A writes, and the paper that computational biologist B writes, and C and D turn into software modules that plug together as easily as Lego blocks plug together. So what is possible with this approach? Well, so you might say, hey, that's great, but where the proof would be in the pudding. Is this just a fantasy? Is this a theory, or is there some reality that's happening here? And so this is an example, and Lenny mentioned my my new license plate, which is really quite excellent, and is really derived from my love of green worms, and this movie really captures, I think, that love more than anything. So what we're showing here is a movie that comes from the Open Worm project that we've been collaborating on and we've been helping guide. And this is a physical description of the way that the best studied organism in all of biology, the C. elegans worm, how it crawls. This is a starting point. This isn't yet complete and by any means. But what's exciting here is this is an example of an integration of a description of physics that encapsulates not only a crawling behavior, but also the forces that are acting on little tiny muscles that are held inside of this green shape that you see that's crawling here, and the interactions between the elastic outsides of this model and a fluid that's inside a little chamber that this guy is crawling in. So this is sort of a closing of the loop from observations that have occurred over many years of the anatomy of the C. elegans. Where are the muscles? How is it shaped? What size is it? Equations that have been derived to understand how does it crawl over, again, many years. Understanding of how force of muscles translates into pulling on a physical shape, and then an implementation of that in computer code. And the other exciting thing about this is that this project is currently being produced completely in an open source and collaborative way. So people from around the world are coming together to work on this and to extend it beyond where it is right now. And like I said, this is by no means complete. In fact, there are many more steps to go with this. But one of the things that's important about this, of course, is that it relies on collaboration and that it relies on software platforms that are able to assemble together all those different kinds of systems of equations that I was talking about before. Okay, so before I sort of show some examples of the kinds of models that we're talking about here, I will kind of come back to this idea of an ideal workflow, of what we'd like to see in a future world and what we're helping to build in computational biology, where we basically want to start with folks forming a hypothesis about what might be going on in a system. Maybe it's in a disease, maybe it's a particular biological mechanism, and then we want them to be able to grab biological entities. And this may be a little abstract to you at first. I'll say a little bit more about what I mean here, but by grab biological entities, I mean that we should be able to essentially pull off of a digital shelf everything that we know about a biological mechanism or a biological entity, a thing in biology, a cell or a tissue or a system. We should be able to pull it off of a virtual shelf and look up everything that anybody's ever said about it and anything that everybody knows about it. And then we should be able to say, all right, well, show me what an example of a model would be that maybe takes a little bit of what's on column A and a little bit of what's on column B and column C and mix them together and have a custom model that now is derived from these various things flying off of our virtual shelf. And then we'd like to be able to explore the dynamics of those things in a very flexible way, and then go back and form more hypotheses and maybe grab some other biological entities, maybe change the ones that we have, maybe tweak our custom models. And the quicker that we can go around this loop, the quicker that we can iterate our understanding of biology across many different domains. So this is the premise, and let me show you some examples of how we might go about making that possible with some of the software that we're working on today. So let me take as one example systems biology as it applies to multiple sclerosis, which was one of the focuses of Orion in phase 1, was looking at the disease of multiple sclerosis. So mostly what we find in the computational field, if you go through the literature, you find models of axons that focus on various parts of biophysics. We don't find any truly integrated models of multiple sclerosis as such. We find that many people are recommending multiscale modeling, and obviously it's difficult, but the importance of it is found throughout the literature. So let me talk a little bit about a couple examples of models that you find to just kind of paint a picture and make this a little bit more concrete. So here we're going to go, I mentioned at the beginning, it's a multiscale problem. So here we're going to go across a couple scales from the physics of axons to the underlying physics of some select molecules that drive the dynamics of axons. So why axons? Well, just as a quick level setting, so multiple sclerosis is a disease that primarily impacts white matter in the brain. The white matter is all the connective tissue, the neural connective tissue. It wraps neurons, and you can find white matter is making up a large volume of the central nervous system in humans and in mammals in general. And what all that white stuff is the yellow stuff over here, which are the myelin sheaths that make up axons. And so a lot, as you can see, because it is the stuff along wiring. This is what I mean when it's sort of connecting different areas together. It is it's showing you how much connectivity there is inside the brain to see where all the white matter is. So in multiple sclerosis, what happens, and we don't exactly know why, but we know that it is a symptom, is demyelination. So we know that the white matter is decaying, it's breaking down, and that's specifically realized in the breakdown of the ability to send messages from one end of a neuron, say its cell body, to distant neighbors that are somewhere over here. So what you start to get in a picture like this are little blotches of lesions that break down and prevent communication from one end to the other. So we can model that, and I already sort of showed you a cartoon of this earlier. We can model that by using what we know about how neurons communicate. They communicate by changing their electrical properties from one end to the other, specifically the membrane potential, the potential across the membrane of the cell from one side to the other. And in a healthy neuron, the one side sends a message, an action potential, and it propagates all the way down through the myelin sheaths to the other end without a problem. When the myelin sheaths are absent, there can be a myriad of problems that occur. For example, you can send a signal and it's not received at all. That's failed conduction. In some cases, you can get a recovered conduction where it's there, but there's some delay that's been introduced, and so it's there, but not as quickly as it should be. There could be evoked after discharge, meaning the message comes in, and then there's a lot more messages that are produced outside than there should be. And then there is the possibility of not even after sending a message in, you get messages that are kind of coming out both ends. And so this is now no longer a communication medium, but it is just creating a lot of noise and a lot of disruption. And these are all things that we find in investigating the neurobiology of multiple sclerosis. So as I said before, you can create a model, and this is an example of a paper that does create a model that looks at the physics of how that works and models these different scenarios that allows us to now get some insight into the disease that then perhaps we can look further into by delving into the underlying mechanisms, such as the ion channels that are affected, or how ion channels are affected by this, which then gives us a protein target that potentially we could target with a drug, or perhaps another kind of therapy we could target as a result of a better understanding of this. So if we dive down now one level deeper, again, I showed you this cartoon earlier, a little at a closer viewpoint. So now we're looking at what was in yellow before is in purple, and looking at the myelin sheets and actually seeing that there's a lot more structure than just a simple cylinder. There's actually many layers of a myelin sheath, and it's wrapping an axon that has many different parts. So we can investigate that as well. So this is an electron micrograph of an axon that has a myelin sheath wrapped around it. So this is now a side view that's cut away here. So the axon's going through the middle, and we're looking at the top part of the myelin sheath and the bottom part of the myelin sheath. And of course, there's these gaps between the myelin sheaths in between called the nodes of Ranvier, which have different electrical properties than what exists within the node. And this is now looking at cross-sections B, C, and D are in here as well. And so we can now schematize that at cross-section and start to also build a computational model at a deeper level that now tracks individual movements of molecules as opposed to equations that approximate movements of molecules as just single variables. So in this paper here in 2008, collaborators modeled both sodium and potassium, as I said, tracking individual sodium and potassium molecules as they diffuse through space in a cutaway. So here, this gap here is the node of Ranvier again, and we've got a myelin sheath on the right and a myelin sheath on the left, and that's in both of these pictures. What we're seeing on the left-hand side, this picture is about sodium, and the one on the right is about potassium. And what they're able to do with this model is to, again, look at how, just understand the complex relationship between how the channels open and close over time, how they are related to each other, how they produce a conduction that passes through this little piece of axon, and ultimately how it relates back to the model that we looked at before. Because in fact, in this study, in the dotted line, the black dotted line here, they're showing an example of the cable model, which is the first model that I showed you before, and in blue, red, and green are examples of the diffusion-based model that I was just showing you previously. And so this is an important thing, this is an important principle here as we're talking about multiscale models, is that they have to sync back together, and they have to be able to have some congruence so that you can now assemble what we've learned at a lower scale and at a deeper scale, which, by the way, is more computationally intensive, and something at a higher scale. So this now allows us to see that we can have a reasonable intuition, at least around this set of parameters, for what's happening at higher scales that's sort of given some ground truth by the simulation at lower scales. So in this way, we start to build up intuitions about what's happening in the actual underlying biology so that we better understand the mechanisms of disease and then go to the step of understanding how we can treat it. Okay, so just another kind of couple other things that happen throughout drug discovery that's related to digital biology, biocomputation. So we know that the drug discovery pipeline is an extremely long, painful, and expensive process. Often there's a lot of outsourcing of this process to labs that use sort of ad hoc software in order to help to do screenings and this sort of thing. Often there's a practice of, in order to speed this up, of simulating the way that small molecules can interact with a particular protein, predicting how small molecules might interact in the desired way. This helps to identify thousands of compounds to test experimentally. And another thing that happens in this field is establishing and evaluating dose concentration response relationships, and subsequently describing and predicting how those time courses are affected by a drug dose, known as pharmacokinetics. So all of this is happening in this general field. And what we're working on at Medicell is the combination between what's currently happening in the field and this more bottom-up explanation of biology, to give you to not only know what a drug will do as it's related to the body, so centered on the drug, but also centered on, well, what does the body actually do, and how does that work? All right, some other areas very quickly to kind of cover that are also applicable areas of modeling that we can address in the same philosophy and in the same spirit. So systems biology as a broader topic as it relates outside of just drug discovery, there are some interesting examples. So the biomodels database is something that's hosted at the European Bioinformatics Institute, And it's a free and open source database that stores quantitative models of biological interest, and it's available to anybody. Currently, they have 143,000 models, and many of those are described in a standard format. 963 of them are published in the literature. The standard that's used here is the Systems Biology Markup Language. And Geppetto, this platform that we've been, as I mentioned, that we've been working on, and we've been collaborating on, is a platform that could potentially allow us to seamlessly interface with the biomodels database, to allow us to run those simulations dynamically and use those models to make predictions. Now, I mentioned this as a separate topic because the models that are stored here in biomodels are not necessarily just about any particular thing, any particular area of disease. They're about biology in general and any kind of biological mechanism. So it might be something about what a microbe does, or it might be something about what happens in a mouse, or what happens in a plant, or this sort of thing. So this gives you a bit more breadth than just an individual focused area. This is an example of biomodels database, a screenshot from a specific model that you can get, and in this case, looking at some pharmacokinetics examples. This particular one looks at interleukin 21. Okay, and so from aggregating models like this together, one of the things that has been one of our interests that we're targeting and we're having a lot of conversations about is this idea of building up a virtual cell. So there have been quite a few exciting works in this space recently in the last few years, publications coming out looking at microbial cells and closing the loop on simulating all of the processes that they can find within a Mycoplasma genitalium, a very small microbe. And what's exciting here as well with this idea of a modular approach is to have a set of modules that describe the different processes that perhaps we pull from SBML, perhaps we work together with partners to describe, and each of those pieces and parts slot in, again, in modules in parallel with each other and allow us to describe the function of an entire cell altogether. So in this way, again, we're solving the multiscale problem by dividing biological processes up into different code modules. Okay, so those are a couple examples of, sort of a tour of some of the modeling space and some models that are existent right now. Now I want to talk a little bit about this grab biological entities thing that I kind of alluded to before, and this idea of virtual shelves and pulling things off of them. So, so for this topic, I sort of want to start by asking ourselves how we ask questions in biology. So maybe we have a question in biology where we'd ask, okay, what are all the cell types in the nervous system that are affected by a disease? And when scientists have that question, today, obviously, we have tools like search engines that we can turn to, or we can use search engines that are a little bit more specific, like literature searches. And we can start to rifle through, perhaps, reviews that hopefully have been written to address this particular question, that is a survey of cell types affected by the disease of my interest. And if that paper is written, then great. If that paper is written but it's 20 years old, then, well, maybe it needs to be revised. Maybe that paper hasn't been written at all. And in fact, the answer is not just in a single paper. The worst case is when that answer is not in a single paper, but it's actually spread across 20 papers, and maybe worst case even still is that only a partial answer even exists, because that's the state of the field. So that's one example. And once you find out what you think is the answer, how do you know and how are you sure that that's the right answer? Okay, another question that we struggle with thinking about how we answer. Or what are the biological mechanisms by which those disease cells malfunction? Another one, what mechanisms of the brain work differently when those disease cells malfunction, and how? And I pose these questions because in an information age that we live in the 21st century, our ability to answer these questions efficiently is far beneath where it needs to be, and far beneath where it is in other domains. We can ask very structured questions about all the books that appear in the category science fiction by going to Amazon, but our ability to answer these questions about diseased neurons is harder. But we are making progress on this, and some of the work that we've done really speaks to this. So So this is where the concept of a knowledge space is introduced. And this is one that we're very excited about, we've worked on in the past, and that there's growing steam behind in the field to have a single place that integrates all this content from around all these papers, so that essentially we can take something like the collection of all neurons that are affected by a disease as a digital object that we can do something with in the modeling domain, that we can plug into a model. So a knowledge space, the idea of it is that it is the repository of all of that knowledge. So in a single place, in a single software application, we have lists of software resources, we have parts of the nervous system, including neurons, brain regions, biomarkers, we have functions of those neurons, the genes that relate to them, the molecules that relate to them, models of those neuron systems, data sets, diseases, and so on. And they're not unrelated to each other. They're in fact put in a way where relationships between them are also captured. And in addition, we should be able to work with that in a digital way, in a way that's befitting of the information age that we're living in. So what might that give us? So I spoke at some length about the multiple sclerosis example. So as we were looking at those pictures of anatomy, there was a lot behind those pictures that are summarized graphically for you and I to look at. But that's not anything that we can plug into a digital framework and start playing with. Those pictures by themselves are just, they're just pixels. So the knowledge behind that, right, that we really want to pull out are details of how do mitochondria work inside those cells. How do those mitochondria interact with other processes? So subcellular processes that we might care about in disease. We might want to specifically pick out just those myelinated neurons in CNS and PNS. We might want to pick out the glial cells or the T cells. And then we want to know all about what's happening between the neurons and the oligos, and between the T cells and the oligos. And facts about these things exist in the literature, and they're littered and spread throughout the literature. And today, it is possible for us to learn about these things in an ad hoc manner, and there are, of course, many companies that offer the service to do informatics research on these things. But to have a resource where all these things are just sitting there and ready to be taken advantage of, this is the idea of a knowledge space. And so principally the way this is all organized is in the context of a semantic graph using semantic web technologies that allows for data to be represented abstractly and connected together using nodes and links. That lets you explore the connections with data much more easily. So all knowledge is essentially nodes and linkages, and that's it. There's no other fancier thing on top of it. That's what happens after you pull your knowledge out. And so if you're not familiar, with graph-based, representations, these are things that you find. You've probably heard of the social graph that's used in social media. So in this case, rather than, it's the graph of your friends, it's the graph of how, say, a neuron relates to a brain region or how a neuron relates to a molecule. So this allows you to do things like pull content. This approach lets you do things like pull content into mashups. And so this is an example from some earlier work that we've been involved with called neuralex.org, which is one of the leading repositories of this form that are out there. And this is an example of a single table view that pulls together information about anatomy, about the cerebellum, across 12, 15 different pages, and puts them into a single aggregated view that shows you a notion of hierarchy, what pieces are under which, and each of these are all links, hyperlinks that are jumping off points to each of those different ideas, which then you can take and turn into a machine understandable description. And furthermore, what you can do with this information is ask for dynamic reports of all the properties that you might find of neurons. So everything that's green here is a place where you learn about a neuron. So the rows here are names of neurons, and across the columns are properties that a neuron has, like their definition or their synonym or their defining citation, or what's the size of their cell body, or if they have spines. So you can very quickly ask across all of this knowledge, what do we know and what do we not know, and in addition, what the actual value of that is. Like, what organism does this particular neuron come from? And this, as you can see, is a much more straightforward way to go about asking the question of, how many neurons have a property, or how many neurons are associated with a disease, than searching through the literature and reading a bunch of papers. All right. So, okay. So Okay, so now that we've talked a little bit about a knowledge space and the idea of a knowledge space, let me say another couple words about models to add on to what I said before. So you'll remember that earlier I referred to the process of going from code that's written up in papers and moving to software platforms that's robust. And why do we care about that problem? Why is that problem important? Why is that something that we as Medicel focus on? And why do we think that it's something that's a problem that needs to be solved? Well, one of the main reasons is that there's a lot of talk these days about reproducibility. And reproducibility is not a problem unique to biocomputation, but it is in fact all throughout biology, but it's also something that computational biology has a problem with. And so let me just demonstrate a little bit about why that is. So here's a paper that defines, that did a survey looking across a bunch of computational biology papers and asking how easy or hard is it to reproduce the results that are found in a typical paper. And they devised what a typical paper looked like, and they went through it and they found all these different steps of things that they had to install or things they had to set up or things they had to understand. And they found that just to reproduce one paper, just to get the answers out from a single paper, took 280 hours, right? It's a huge, huge time investment for something where essentially there's already computer code that's been written and that had run on some computer, right? So that, in a single picture, is an example of why this challenge of going from a paper to software platforms is so vexing. So now let me sort of, that's sort of the setup to say, why do we need better and more robust software platforms to help us ease that transition and help make that process a lot easier and make it more like point and click. So this is where we segue to Geppetto. So Geppetto is a really terrific community-driven project that we've had the benefit of being a part of. Now there's actually a lot of people contributing to, and that we think is really, heading in the right direction to help solve this problem. So it's an open source simulation platform that's aimed at multi-algorithm, multi-scale simulation of biological systems in their environment. This is what it looks like. What I want to emphasize about this picture is that this is entirely self-contained inside of a web browser. So this is a platform for examining models in computational biology that's really built for the 21st century, that's built for Web 3.0. So rather than a long, complicated 280-hour tool chain to get a computational model working, it's a URL that loads in any browser in the world. And in this case, we see in the background a sphere that's a three-dimensional representation of a model. In the foreground, we see graphs that are generated from crunching the numbers and crunching those equations that go into describing, in this case, a Hodgkin-Huxley spiking neuron. Just one example of some mathematical physics that is core to neuroscience. You're able to load an arbitrary simulation from anywhere around the web, and you're able to see the results of it here in your web browser and explore them with a dynamic console. So this is sort of built, and this again addresses the challenge that what we find is that the code that comes from papers in academia, which is of course very important and provides a lot of great insights, but it's built in a way that from the perspective of software engineers is not optimal. From the perspective of how industry looks at building code, they look at building code like Lego blocks. They want to build components that snap together. But often every single paper is disconnected from every other, at least is the current state in the last sort of 20 or 30 years of computational biology. So this Geppetto project is really a partnership of lots of people coming together and say, hey, actually we can learn something from the way that industry does this, and we can start creating our models in a way that's better than what we had in the past. So what does it do? It provides a web application that controls the simulation through the web browser. It provides a way to define what needs to be simulated in a data-driven way. That's really important because all too often in the past, computational models have been written such that the data and the code are the same thing. And another important lesson to learn from industry is to separate those two provides you a lot of benefits. It defines interfaces to allow third-party simulators and model interpreters to extend the platform. So all too often, tools that have been produced provide limitations that cause people to want to work around the tool. So extensibility is a really important criterion for anything that folks are going to work on collaboratively together. It provides a way to stream the simulated data from the back end to the front end. That was shown by the graphs before. And it provides a 3D engine to visualize in the browser the stuff that's being simulated. So more and more often, spatial considerations of computational models are taken into account here. And so what's really critical is that this now provides a new way to think about collaboration on models. Is that rather than sending you a PDF paper that takes you 280 hours to unpack, you're sending a link that allows people to see what's happening. The kind of collaboration that's available is really a quantum leap forward. So having access to your simulations from anywhere in the world through a web browser is really a whole new paradigm for looking at computational biology. It's a way of visualizing and interacting with models that doesn't involve installing any software at all, well, except the web browser. Most people have that. And it creates the ability to model complex systems that require using different simulators and algorithms. And so let me say a little bit more about that. So the architecture underneath it, I mentioned before this modular approach, that we really want to take what comes from one paper and put it in one code module, and what comes from a second paper and put it in a second one, and have them all add up. Well, that's doable, and there's many parallels in industry where this is done in different domains, in different domains of engineering, for example. It hasn't been so much done in biology, but the Geppetto platform is one really where, again, a lot of people are coming together and saying, yeah, actually, this is an approach that we want to take. So it takes some engineering. So it takes, for example, having standards for defining those data-driven simulations that can be read by a core layer. It takes an integration layer that can deal with pulling these different modules together. And then it requires engineering a system that's able to work on the web and able to work with multiple clients and able to respond to multiple hits. And it needs to be able to give you the nice features that you have in any advanced modeling system, like a console, the ability to plot, and different kinds of pop-ups and tutorials. So I realize that I'm going short on time here, so I'm going to skip the demo for the moment, but I do want I will point you at this to have a demo of your own, and maybe if there's time at the end, I can do the demo. So I just want to make a couple more comments about this. So how is it being developed? So it is in fact open source under the MIT license. It's a growing community that's the product of 2.5 years of coding, of 10 active contributors currently, and more who are contributing little pieces and parts throughout. There is a bi-weekly Google Hangout that's open and anybody can join. You can find out more about that at the website. It's an open process for defining how it works. So really, although I'm presenting this today to you in the context of a presentation that I'm giving in Medicell, this is really a much larger community that goes outside of us, and we don't make all the decisions in how it works. It has regular monthly releases, and the latest has been 0.1.5 alpha. And to just kind of underscore this open development model and the way this works, this is a picture of the way that this is a picture of the tasks and the way that tasks are defined for how we're going to extend that platform together. So every improvement or bug to the system is logged here, originally in GitHub, and then this is a kind of a cover over GitHub. And every 2 weeks, folks get together and decide what they're going to do and how they're going to add to this and work on it. And this is the idea of a Kanban board where cards are moved from a to-do to an in progress to needs review and eventually over here to done. So already in the field, we're seeing this collaboration on an engine. And why do people do this? Why do people contribute? Because they have exciting models that they are building that require a platform like this. And so in addition to collaboratively building the platform, there's collaborative model development that's happening here. So you can go check this out today at live.geppetto.org. And if you hit that, you will be greeted with a welcome screen that will ask you if you want to go through a tutorial. Please have a look at it and give us any feedback. There's the ability to give us feedback in the bottom right-hand corner that this whole community can share. You can visit the website at geppetto.org. There's a documentation site at docs.geppetto.org. And you can also visit the underlying developer section for the code folks amongst you at github.com/openworm.org/org.geppetto. So where is this going in the future? We have a in the next 2 years, what this community is aiming at is the ability to build on top of this environment increasing numbers of standards that include potentially SBML that I mentioned to you before, NeurML, some other languages that are in the panoply of standards that are in this field, to increase the support for different mathematical equations and models, ordinary differential equations, partial differential equations, and so forth. The ability to do hybrid systems and stochastic systems. And then additionally to extend to different domains such as synthetic biology and drug discovery. So at the end here, what we have is this picture of a platform that's being collaboratively developed, and to just kind of tie it back to where we began with these different models, for example, of MS, what we want to have is a front end where instead of taking the long road to exploring, to finding a model in a paper, is that this model here appears on the same web-based platform as a model at a different scale, and you can look between them, and they all appear here in the same level of abstraction and simulation. And ultimately, then, these models should be described by what we get out of the knowledge space that we talked about before, so that not only do we have the models, the computational models that have been described in the past, but also the knowledge that went into those models that define for us together what the underlying biology was that we're in fact simulating. Okay, so that is where I will stop and take questions from the audience. Let me get back to the Hangout view here.
Hi, Stephen. I do have, this is Jeremy. I do have one question to your point of creating the knowledge base and then the analogy to a search engine or something like Google. I was curious, where are a lot of those, the information, the results from those search engines coming from? Is it, are there government entities and databases there that it's able to tap into? Is there a network of universities, or is it really up to individual researchers? And, I saw that your point of the MIT collaboration, but, kind of how, to get to that Google level of the search engine, how widespread is that network?
Right, so how does the knowledge get into the knowledge space, essentially, and where does it come from? Work has to go into fitting all that knowledge together, and the original sources can come from sources like you mentioned. So a lot of databases that have been publicly funded, for example, their data are being integrated into knowledge spaces like this, or Neuralex.org in particular is one example of this. But in addition, the other aspect that I didn't talk a lot about was that a knowledge space is something that's in fact editable by the community. So much like Wikipedia is a knowledge source that anyone can edit, a knowledge space is something that anyone can edit, but at the level of a description of the biology that's computational and that machines can read, not just humans. So it's both the combination of folks who care about giving new life to data that was generated from a publicly funded resource, as well as individual scientists who maybe find an error and want to fix something, or find something missing and want to add that back in.
Okay,
all
great.
right.
Thank you.
Great. Stephen, I've got a couple of questions. One is on the group chat from Magali, and I'll just read it to you. You can probably, can you see it there, or do you want me to read it to you?
Okay. Looks like thanks for a great presentation. Thank you, Magali. You gave one example of an MS model. What are the greatest obstacles to doing bottom-up modeling for human brain disease? What are the greatest obstacles to doing bottom-up modeling for human brain disease? Well, one is that our challenge of bottom-up modeling for human brain disease is related to the challenge of bottom-up modeling for human brains, which is that as we understand the physics of the brain. As I started with my presentation, first we understand the physics, then we turn those physics into equations, then we turn those equations into code. So the physical understanding of the human brain is already by itself a challenge today. So our physical understanding of the human brain starts with what happens within individual cells, and so that's already a problem because there's so many of them in the human brain. So now we're dealing with very large numbers of different parts that we have to track. And then, if that wasn't bad enough, we have to deal with all the interactions between them. So we're talking about 10 to the 12 neurons, 10 to the 15 connections, and all of them we have some clues about the physics, but our physics is still, our understanding of the physical nature of that tissue is still limited by a lot of our experimental methodologies. So one of the greatest obstacles to doing that is to fully unpack all of those physics and turn them into computer models. Okay, but a lot of folks seem to have taken the approach that until we understand all of the physics, we can't start with any of the modeling. And I also don't believe that that's true. And I believe that there are a lot of ambitious efforts that are really blazing new ground for working with the data that we do have today. Because in fact, I think computer models can help us in the understanding of physics by starting with the little physics that we do know, assembling them together in small packages of what we do know, and then using the computer to help us ask questions at higher scales. So we can start with knowledge that we have about one brain area and one small network of neurons. And then we can say, well, what if we paste that together with 2 or 3 or 4? And does that match what happens when we measure at a larger scale? So I'd say a second obstacle to bottom-up modeling for human brain disease is to come up with the right strategy for how you assemble what physics you do know into the right models that then give you insight into the disease, or that give you insight into the normal functioning, and then the disease is probably the next step, which is to say, okay, now I think I understand how it works correctly. I have a general understanding of that. Now I see when I measure when I measure cases where the disease is present and apparent, that now this function that I seem to understand well how it works correctly, how is it failing? And now that next step is to ask additional questions about making hypotheses about, well, what are the physics that are underlying that disease? Is it because myelin sheaths are degrading? So why are myelin sheaths degrading? What is attacking the myelin sheaths, or what are they not getting that they normally have? And so the third obstacle is in unpacking that and to build models that are expressive enough to let us even get in and start asking those questions and get us into a fertile brainstorming place where we can really very flexibly say, okay, I'm going to turn off all the myelin sheaths and see what happens, or I'm going to turn them all on, or I'm going to double them, and does that do something, right? So I'd say that we're just now building some of the frameworks to let us piece together the little physics that we do know into models, and we're just starting to piece together little pieces of the physics of disease and put those into models. So I think that ultimately, and bring it back to where we are, it's this challenge of integrating what we know into solid platforms that's really at the core of bottom-up modeling today, and one of the challenges that we really need to overcome before we can go to the next level and do some really exciting things with the data that we have.
Great, thanks, Stephen. You have another question from Alden Masonis. Can you optimize establishment of translational models by interrelating overlapping semantic maps?
Okay, can you optimize the establishment by interrelating overlapping semantic maps? Yes. Well, okay. Optimize assumes like that they're perfect. Maybe. Can you improve them? I think the question is, can you improve them with semantic maps? Sure. Right. Okay. So let me unpack what that's saying. So a translational model is something that goes from what the scientist measures to what we can make a product from to hopefully cure a disease. Right. So that's a translational model. Can that benefit from semantic maps at all, first of all? Yes. So a semantic map is just a fancy way of saying that we took what we think is happening with something and we put it into a computer, and we have a means of asking that computer to tell us again what we told it, and hopefully tell us what we told it from maybe a different angle, so that we can actually get some new insight that we couldn't see before. So an example of this is that, like, we spend several months telling a computer everything we know about 100 neuron types, and we know everything about those 100 neurons, and then at the end of those 3 months we ask one question that summarizes everything about those 100 neurons in some new way. Okay, so we told them about 100 neurons, we said what the neurotransmitter was, and now we're going to ask a new question, which is, I want to know, of all those 100 neurons that I told you about, which ones have glutamate as the neurotransmitter, and which ones of them are in a particular brain area of interest. And although you kind of knew that to begin with, the ability to flexibly look at it from a new angle is very powerful. So a semantic map gives us the potential to do this more and more and more as we build it up, so that we have descriptions not only of the types of neurons, but what's going on inside them, how their organelles are interacting, knowledge about that kind of thing. So certainly I think you can improve translational models by being able to ask the computer complex questions about knowledge that that you didn't have before. Otherwise, your only resort is to go to paper, or hopefully to find the one database that talks about the thing that you want to ask. But more often than not, we have multiple databases that overlap a little bit, right, that we can't ask questions between. So we've got a database of connectivity and a database of cell types and a database of mouse models, and they don't talk to each other, and there's no way to pull them together. So the semantic map is a tool to let us take everything that we know from those different databases and put them in the same space of query. So yeah, absolutely. I think the translational models can certainly be optimized, improved, by interrelating overlapping semantic maps that come from pulling the knowledge of these different databases together. Yes.
Great. And the final question is, and you've got about 2 minutes to answer this, and then I'll sign us off, is how do you see this effort in relation to the very ambitious European Human Brain Project?
Well, they're not entirely different. I would say that that there's a lot of shared philosophy between what we work on and what the European Human Brain Project is working on. They also care about taking an approach that marries together software engineering and an industrial approach to solving problems with modern neuroscience. So they care very much about the effort of dealing with data within neuroscience. So there's a philosophical and methodological overlap with what we're doing and what they're doing. Now, the differences are they're a research project, so their aim to come out of this, their end to come out of this, is a better understanding that goes into scientific knowledge. And in our case, our aim is to help other people, help other organizations do the most that they possibly can with the papers that exist and the models that exist today. So we're not trying to do new science, really. We're trying to take science that exists and package it up and give it to people in a way that's convenient and packaged so that they can actually use it in a flexible manner. I think that's really the effort that we see as the real challenge that we're trying to solve. So I'd say that they're complementary. I'd say that they have shared philosophy, but they are obviously different in our ultimate goals and our ultimate aims. So I'm not trying to solve, an understanding of the human brain myself, but I do want to contribute to the tools and infrastructures that are related to that effort.
Great, and we have one last question that just slid underneath the door.
Sure.
You want testable hypotheses. For MS, for example, how many axons die before you lose brain functions or muscle power? How is the rate of decay for a relapsing remitting patient? What factors affect those calculations? And do you see these simpler questions?
All right. Yes. So I agree. So you do want all these testable hypotheses. I guess I say I wouldn't disagree with any of those. I would comment that the way that hypotheses are formed today are all in our heads. So we ask these questions and we communicate them to each other and we talk about them. But often the challenge is that there's not a lot of underlying rigor to help us understand those. So how do we go about assembling those hypotheses in an even deeper way? Is often a challenge that we find. So we may ask this question like how many axons die before you lose brain functions, but there's no way for computers to help us answer that question so long as they just remain either in our heads or so long as they remain just in the subject of communication or in the subject of written up in academic articles. So instead we can start to assemble we can start to flesh that hypothesis out. So we can start to say, well, how might we answer how many axons die before you lose brain function? So what are the patterns that we see of where lesions occur in MS? Is there any data in that? Okay, so once you have that, then you can say, well, can I put that in some anatomical context of the rest of the brain? If I can, then I can now start to assemble things that we know about other parts of anatomy. So maybe we know that there are particular cell types in the parts of brain that are more likely to have axons die, and maybe we can assemble more knowledge. So at the point that we just communicate it verbally, it's hard for us to bring to bear everything else that's being talked about and learned throughout neuroscience. But at the point at which we can turn those hypotheses into computable questions, now suddenly we open up and we unlock a vast amount of information that is being put out into the internet, on into the scientific world every day. And we're never going to be able, basically, to read thousands of papers, but we can use a search engine that has computationally, digitally read all those papers, or that's the assemblage of a large amount of collaborative effort to kind of package that and pull that together into something like a graph where those questions can be asked. So I agree. All those are important. All those are important hypotheses. And then what comes out at the end of that? Yes, of course. Once now you've gotten a very clear and specific hypothesis, then yes, you need to take that. You need to take that and you need to make a clinical trial, and you need to see if that is in fact the case. You need to have ground truth data at the end of the day that suggests that. But often we can't even imagine what the best questions are to ask because we're still sort of lost and trapped in this what appears to be a paucity of data, but in fact is a paucity of interconnections between data at the end of the day. So, but yes, I would also agree that at the end goal of all of this modeling, testable hypotheses are critical, and they're in fact the whole point of doing this. But to get there today is still a challenge.
Great, thank you, Stephen, very much, and thanks for everybody for dropping by. I think you just landed on the topic for your next talk, the paucity of interconnections between data. I'll now just do a brief plug for our next webinar, which will be coming up actually in just a few weeks in August, on August 7th, Thursday, same time, same place, same Orion Bio Network, featuring Netazak, PhD, the Chief Science Officer at Prize for Life. Her topic will be ALS patients being proactive. Can ALS big data bring the next ALS breakthrough? So look forward to seeing you all and all your friends for that. Again, thanks, Stephen. Thanks, everyone. And Orion Bio Network signing off.
Okay, thank you.